Hello everyone. So welcome you all again to my YouTube channel Let's Do Biology. I hope you all are doing well. So I know कि मैं ये वीडियो बहुत बहुत टाइम बाद बना रही हूँ because I was not getting any time with my like after my PhD work. So myself Archana and I am research scholar at IISc Banglore. So finally you all are getting your exam after some days for NII. So I thought I should make a video for something like that can help you. So मुझे students से ही अपने and also like if you haven't joined yet to my Telegram group, so you can join me on my Telegram group. I will provide the link in description box. And guys, if you haven't subscribed yet my YouTube channel, and then please subscribe my YouTube channel. Let's do biology and do share with your friends also. ओके सो ये क्वेश्चंस मैं जो डिस्कस करने वाली हूँ इस वीडियो में ये इसी ईयर जो अप्रैल में एनआईआई का एग्जाम हुआ था उससे रिलेटेड है. So I got some questions only from the students on my Telegram group itself. So I will discuss only those questions and also जो भी क्वेश्चंस होंगे उनसे रिलेटेड उस टॉपिक को भी मैं अच्छे से डिस्कस करने का ट्राई करूंगी सो दैट यू विल अंडरस्टैंड दोज ऑल्सो. ठीक है. सो अच्छे से सुनना एंड वीडियो को स्टार्टिंग से लास्ट तक पूरा देखना. एंड मेरे चैनल पर आपको एनआईए से रिलेटेड बहुत सारी वीडियोस मिल जाएंगी. लाइक इफ यू वांट टू वॉच सिलेबस रिलेटेड वीडियो देन एनआईए सिलेबस का वीडियो भी मैं ऑलरेडी अपलोडेड कर चुकी हूँ. एंड ऑल्सो पीवाईक्यूज लाइक लास्ट ईयर का पीवाईक्यूज भी है और एक और लास्ट ईयर का पीवाईक्यूज भी मेरे चैनल पर है. देन दो तीन कुछ प्रैक्टिस सेट है उसमें कुछ क्वेश्चंस है जो मुझे लग रहा था कि इम्पोर्टेंट है तो जिनसे आप प्रैक्टिस कर सकते हैं वो भी मेरे चैनल पर ऑलरेडी अपलोडेड है. ठीक है. सो लेट्स स्टार्ट द वीडियो विदाउट डिलेइंग. ओके. So this is the first question. What is the question? Postprandial increment in plasma glucose levels decrease by which action? So in this you have these four options. So in this question what they have asked कि ग्लूकोज लेवल जो है जब पोस्ट प्रेंडियल इंक्रीमेंट होता है उस टाइम प्लाज्मा में ग्लूकोज लेवल डिक्रीज किससे होता है. तो इसमें डिफरेंट एंजाइम हमें दिए हुए तो हमें बताना है ग्लूकोज का लेवल डिक्रीज कब होता है कौन से एंजाइम से होता है. ठीक है. तो आई विल ट्राई टू एक्सप्लेन दिस इन एज मच एज डिटेल जितना पॉसिबल हो पाएगा. ठीक है. सो फर्स्ट ऑप्शन है ग्लूकागन. तो ग्लूकागन तो यू ऑल नो कि इट यूजली इंक्रीज द ग्लूकोज लेवल. ठीक है. इंसुलिन और ग्लूकागन ग्लूकागन जो होता है वो इंक्रीज करता है इंसुलिन जो है डिक्रीज करता है. तो इंसुलिन तो इसमें ऑप्शन दिया हुआ नहीं है. ठीक है. देन नेक्स्ट क्या है ग्लूकागन लाइक पेप्टाइड 1 गैलेनिन ग्रेलिन. तो ये तीन डिफरेंट है इनका मैंने फंक्शन यहाँ पे लिखा भी है कि जो ग्रेलिन होता है that can increase glucose level by decreasing insulin. And how it is increasing by affecting glucagon secretion. So it can increase the glucagon level so that insulin will decrease and ultimately glucose level will go high. तो ग्लूकोज लेवल ग्रेलिन तो होगा नहीं हमारा क्योंकि इससे इंक्रीज हो रहा है. तो इन सबको भी याद रखना. ग्रेलिन क्या कर रहा है ये कर रहा है इंक्रीज. ग्लूकागन भी ऑलरेडी नो कि ये तो इंक्रीज करता ही है. देन अनदर ऑप्शंस आर जीएल. पहले हम देखते हैं गैलेनिन. इट इनहिबिट्स ग्लूकोज रिलीज बाय hyperpolarization and lowering cytoplasmic calcium concentration. Okay. And the next one is GLP-1 glucagon like peptide 1. It can attenuate ghrelin action. This will इसका मतलब है कि ये रोक देगा ग्रेलिन के एक्शन को. ग्रेलिन क्या कर रहा था इंक्रीज कर रहा था तो इसका मतलब है GLP-1 किसी तरीके से डिक्रीज करेगा. तो हम वो अब देखते हैं कि डिक्रीज कैसे करता है. ये देखो, ये है GLP-1 जो कि एक्ट करेगा डेल्टा पैंक्रियाटिक सेल में, अल्फा बीटा डेल्टा गामा ये सब सेल होती है, तो ये एक्ट करेगा डेल्टा सेल पे, बीटा सेल पे एक्ट करेगा, सो दैट किस पे एक्ट करेगा, बीटा सेल के GLP-1 रिसेप्टर पे। उसकी वजह से इंसुलिन का सिक्रीशन बढ़ जाएगा, इंसुलिन का सिक्रीशन और बायोसिंथेसिस बढ़ जाएगा। इंसुलिन का सिक्रीशन बढ़ रहा है मतलब कि प्लाज्मा में ग्लूकोज लेवल डिक्रीज हो जाएगा, और डेल्टा सेल पे एक्ट करेगा तो आगे अल्फा सेल पे जो एक्ट करके वो इनिबिटरी एक्शन शो करता है डेल्टा सेल, जो कि ग्लूकागन की सिक्रीशन को कम कर देगा। ग्लूकागन सिक्रीशन कम हो गया, इंसुलिन सिक्रीशन बढ़ गया, मतलब कि प्लाज्मा ग्लूकोज लेवल डिक्रीज हो जाएगा। तो इसका मतलब आंसर हमारा होगा ऑप्शन सेकंड। सो दिस मींस what here what GLP-1 is doing, GLP-1 decreasing the glucagon level and increasing the insulin secretion, so ultimately it will decrease the blood glucose level. Okay, so what it is doing, it is it is increasing the effect or like increasing the secretion of insulin and decreasing or inhibiting the glucagon secretion, so that plasma glucose level will decrease. So our answer will be option B, GLP-1. ठीक है, आई होप ये क्वेश्चन समझ आया होगा आपको. So let's go to next question. So what is our next question? Which method used to delete terminal amino acid? So in this question, they have asked which method, like there are different methods that are used for determination of the amino acid sequence. In this question, like I got only two options, so from these options only I will try to discuss with you. ठीक है. So what we got, Admin, degradation and Sanger. So इसका आंसर होगा फर्स्ट, एडमेंट डिग्रेडेशन. सो व्हाट एडमेंट डिग्रेडेशन डू, इट विल it will sequentially remove one residue, one amino acid residue. It will sequentially remove one by one from n terminal of peptide. Okay, so what it is doing, it is deleting terminal amino acid by one. Okay, and this is Sanger. Sanger is a sequencing method which we use to determine the sequence of sequence of nucleotides actually. Okay, not for the in this we don't use deletion of terminal amino acid, so our answer will be Admin degradation. Now I will try to elaborate Sanger sequencing. ठीक है, so how it is used to be done, this is the sequence that we have. We don't know this single standard DNA, this is there, we don't know its sequence, so we will determine the sequence by using this Sanger sequencing. What we will do, we will set up a reaction in which we provide this unknown sequence DNA polymerase, which will polymerize the DNA, these nucleotide DATP, DCTP, DTTP, all the deoxynucleotide. Then we will provide radioactively labeled primer like DD. So we will prepare different. This is the primer, like which will bind, which will be a small sequence, which will bind to the end, like which will be having the homology or complementary to the sequence. It will bind there. So like it is complementary here, so it will bind there. We will make 4 different reaction mixer by using different dideoxy. What it used to do, when we will provide dideoxy ATP, it will stop the polymerization at that point, because यहाँ पे OH नहीं होगा, it is dideoxy. ठीक है, तो this will stop the reaction at the point when dideoxy adenosyl triphosphate will come. So at that point, the polymerization will stop. In the same way, it will happen for all the 4 nucleotide. ठीक है, then what we will do, we will see कि where this stop is occurring. Like I am telling now, whenever this DDA will come, it will stop here, then next DDA will come, it will stop. Like there will be probability that different type of thousands of nucleotide sequence will form. Then we will see it by running on a gel. So they will run according to their size. Then we will read this sequence according to their base pair, like according to their molecular weight. Wherever these will come, we can determine the sequence of amino acid and deduce the sequence of this unknown sequence. ठीक है, so in this way we can determine the sequence. So अभी मैं इसको समराइज करती हूँ. what we can do in this case, हमारा एक अननोन सीक्वेंस होगा, हम वहां पे प्रोवाइड करेंगे डीएनए पॉलीमरेज, विल प्रोवाइड न्यूक्लियोटाइड्स एंड ऑल्सो रेडियो लेवल प्राइमर, और हम 4 डिफरेंट रिएक्शन सेटअप करेंगे बाय यूजिंग डीडीएटीपी और डिफरेंट अनदर न्यूक्लियोटाइड, सो दैट क्योंकि ये डीडी है तो इसलिए जहां पे भी ये आएगा हमारे सीक्वेंस के कॉम्प्लीमेंट्री, वहां पे आगे पॉलीमराइजेशन स्टॉप हो जाएगा. अब जैसे डीडीए आ गया यहाँ पे तो ये यहाँ पे स्टॉप हो जाएगा. अब प्रोबेबिलिटी है कि ये थोड़ा सा और ज्यादा बढ़ गया और उसके बाद डीडीए आया तो वहां पे स्टॉप हो जाएगा. अब यहाँ पे आगे टी है तो यहाँ पे फिर से ए आएगा तो ये यहाँ पे स्टॉप हो जाएगा. मतलब डिफरेंट न्यूक्लियोटाइड सीक्वेंस होंगे जो डिफरेंट स्पेस पे स्टॉप होंगे. ऐसे ही सीटीपी के साथ होगा टीटीपी और जीटीटीपी. अब हम इनको कैसे रीड करेंगे? ये हमारा सबसे लास्ट में आ रहा है सी, इसके बाद आया जी, देन ए जी, सी, टी, ए, देन अगेन जी. इस तरीके से हम इसका सीक्वेंस पता कर लेंगे और इसका जो कॉम्प्लीमेंट्री सीक्वेंस होगा वो हम डिड्यूस कर देंगे कि ये हमारा जो अननोन सीक्वेंस था वो हमने डिटरमाइन कर लिया. ठीक है, सो दिस इज हाउ Sanger sequencing used to done. So the next question is thiamine deficiency in which disease? So this is asking in which disease is caused due to thiamine deficiency. So मैंने एक टेबल लगाया है. This is the table that is from Pathfinder. ठीक है. सो हाँ, एक और चीज मैं डिस्कस करना चाहती हूँ. बहुत सारे स्टूडेंट्स का ये क्वेश्चन होता है कि हाउ शुड दे प्रिपेयर फॉर एनआईआई. So frankly speaking, students, you can just prepare from Pathfinder itself. You can find one video on my YouTube channel also that how you should prepare, or like is Pathfinder is enough for entrance exams like CSIR, DBT, GATE or whatever, NII also. It is enough. If you if you will read it like a single word, so that you can learn or grasp a single word itself, then Pathfinder is enough for you for clearing or even for ranking top high rank. Then also it is enough. ठीक है. So anything you can find in Pathfinder, but you have to read it many times. ठीक है. So in this case, what they are asking, ये table है एक, so you have to learn it very clearly. आपको इसमें से एक एक वर्ड याद होना चाहिए. फर्स्ट थिंग, लाइक थाइमीन है तो इसका ये कौन सा विटामिन का नाम है. लाइक यहाँ पे आप देख सकते हो कि डिफरेंट विटामिन के नाम है, जैसे राइबोफ्लेविन, विटामिन बी2. फर्स्ट थिंग आपको ये याद रखनी है इस टेबल में. दूसरा, ये जो थाइमीन है और विटामिन बी1 है, ये इसका एक्शन क्या होता है. जैसे ये कोफैक्टर होता है डीकार्बोक्सिलेशन में, तो ये सब जगह लाइक सीएसआईआर, गेट, बहुत ज्यादा बहुत फेमस, और लाइक ऐसा क्वेश्चन है जो आपको हर जगह मिलेगा ही. तो ये टेबल किसी भी तरह अच्छे से रट लेना, कैसे भी हो जाए. ठीक है. तो इस टेबल में फर्स्ट थिंग याद करना है कि कौन से विटामिन का कौन सा नाम है, इसका एक्शन क्या है. तीसरी चीज डेफिशिएंसी. ठीक है. और क्या, वाटर सोल्यूबल कौन सा है, फैट सोल्यूबल कौन सा है. बस ये चीज याद कर लेना. इसमें से आपका एक भी क्वेश्चन ऐसा नहीं रह पाएगा जो छूट जाएगा. ओके, सो लर्न इट लाइक यू कैन सोल्व एनी क्वेश्चन. लाइक अगर आप जो भी वहां पे पाथफाइंडर में बहुत सारी चीजें दी हुई है, इफ यू कांट रिकॉल ऑल द थिंग्स, देन जस्ट ट्राई टू लर्न दिस टेबल. ओके, डोंट स्किप दिस वन. सो यहाँ पे हमसे पूछा है विच डिजीज कैन कोज बाय थाइमिन डेफिशिएंसी. सो क्लियरली इस टेबल से पता लगा सकते हो थाइमिन की डेफिशिएंसी से कौन सा डिजीज होता है, बेरी. सो और आंसर विल बी ऑप्शन 3. देन यहाँ पे स्कर्वी, तो स्कर्वी ढूंढते हैं. ओके, नाइट ब्लाइंडनेस मिल गया, विटामिन सी, स्कर्वी, दिस इज द वन. देन परनीसियस एनीमिया, दिस इज विटामिन बी12. So see, all the options related to this question are also here in this table. तो आपको वो भी याद होना चाहिए. ऐसा नहीं है जो क्वेश्चन में पूछा है बस वो याद कर लो. तो टेबल में जितना कुछ भी दिया हुआ है सब कुछ याद करना. ठीक है. देन मूव टू नेक्स्ट क्वेश्चन. here what they are asking, which pair do, and also like now I am going to talk about this immunology related question. So many immunology topic related videos I have uploaded on my YouTube channel. Like most important is MHC, like antigen processing and presentation, MHC 1 and MHC 2. उन वीडियोस को आई विल रिकमेंड यू कि उनको जरूर देखना, क्योंकि उसमें मैंने NII में बहुत सारे क्वेश्चंस हर साल उनसे पूछे जाते हैं. तो आई विल रिकमेंड यू कि उन वीडियो को अच्छे से देखना, क्योंकि उसमें मैंने सारे पॉइंट जितने भी लाइक एक्सपेरिमेंट हो या फिर उनसे एक एक छोटी से छोटी चीज मैंने इंक्लूड की है. अगर आप, क्योंकि वो वहां पे मैंने क्यूबी को रेफर किया है, अगर आप क्यूबी को नहीं पढ़ पा रहे हो तो आप वो वीडियो अच्छे से देखना, सारे एक एक क्वेश्चन आपके सोल्व हो जाएंगे उससे. ठीक है. सो दिस क्वेश्चन इज आस्किंग विच पेयर डू एंटीबॉडी मीडिएटेड किलिंग ऑफ पैथोजन. ये क्वेश्चन हमसे पूछा गया है. एडीसीसी प्रोसेस होता है एंटीजन डिपेंडेंट सेल मीडिएटेड साइटोटॉक्सिसिटी, जिसमें क्या होता है मैं फिगर के थ्रू समझाती हूँ. ये पाथफाइंडर से लिया है, ये मैंने एक डिफरेंट साइट से लिया है. सो हमारा ये टारगेट सेल है. इस टारगेट सेल पे ये एंटीजन प्रेजेंट है, यहाँ पे हमारा एंटीबॉडी बाइंड हो जाएगा. ठीक है. वेरिएबल रीजन है, वो बाइंड होगा एंटीजन बाइंडिंग में. इसका जो एफसी रीजन है वो फ्री है. अब क्या होगा, ये है एफसी रीजन. ये एफसी रीजन एनके सेल्स द okay, so this fc region is free. Then what will happen next, our NK cells, natural killer cells, our phagocytic cells, in which NK cells and macrophages, these two cells, they can bind to fc receptor of this antibody. And then next what will happen, like in this question, in this figure you can see, after binding to the NK cells to the fc region of antibody, they will release their enzymes. Okay, these cells can be natural killer cells or macrophage cells, which will bind to the fc region. This question is usually asked in entrance exam, that which region of antibody will bind to the receptor of NK cells. So fc receptor, fc part will bind to the fc receptor of natural killer cell or macrophage cell. Then this will activate our NK cells and release the enzymes, perforin and gran enzyme, that will ultimately lead to the apoptosis of the target cells. In this way, this ADCC process occurs, that one will be our target cell, which will be having antigen, antibody will bind there, that fc region of antibody will be free, that will be bounded by fc gamma receptor, which will bind to the fc region of antibody, that ultimately lead to the activation of NK cells. That activation will release or activate the enzymes and release the perforin and granzyme cells, and target cell will apoptize. ठीक है. सो अभी ऑप्शन देखते हैं. अभी आपको प्रोसेस समझ आ गया होगा, एंड आई एम होपिंग. मैक्रोफाजेस एंड बी सेल, नो. B cell doesn't involve in ADCC, no TC cell, macrophage and NK cell, yes. Macrophage and TC cell, no. These macrophage and NK cells involve in antigen dependent cell mediated cytotoxicity, sorry, antibody mediated killing of pathogen, because यहाँ पे पैथोजन का किलिंग किससे मीडिएट किया जा रहा है, मीडिएटर की तरह यहाँ पे एंटीबॉडी एक्ट कर रहा है, बिकॉज इट मीडिएटर इज लाइक समथिंग विच विल इन्वॉल्व इन बिटवीन ऑफ टारगेट सेल एंड आवर फैगोसिटिक सेल. ठीक है. सो नेक्स्ट क्वेश्चन इज टी सेल मैच्यूरेशन इन थाइमस. The maturation of T cell occur in thymus. So I will try to explain this topic whole, like so that you can attempt other questions also. ठीक है. So these are the options. First I will try to explain the topic so that you can get the topic. After that you can answer these questions very easily. ठीक है. So ये मैंने QB से ही लिया है डायग्राम. तो फर्स्ट क्या होता है हमारी, फर्स्ट हिमेटोपोएटिक स्टेम सेल प्रेजेंट इन बोन मैरो, दैट विल टर्न टू प्रिकर्सर सेल, देन इट विल माइग्रेट फ्रॉम ब्लड टू थाइमस. इन थाइमस cortex region it will become T cell precursor. That further from here, this is the early thymocytic development that starting from here. First it will become DN1. DN1 is double negative one. Okay, here nothing will be there on its surface. Then the maturation of the gene starts on the surface of the T cell. Here, okay, this is written as TCR beta, locus rearrangement proliferation. So here TCR beta locus gene will start rearranging on the surface of double negative T cell, precursor T cell. After DN2, T cell will commit either it want to go to beta or gamma. Okay, we have two different type of T cell, alpha, beta and gamma delta. Mostly alpha, beta are the cells that are present in a normal person, but like at the child stage, gamma delta T cells are also present, but these are very minor, like less than 5% only. Okay, most are the alpha, beta. If at this stage, if it is beta, then it will go to alpha beta selection. If it is gamma, then it will go to gamma delta T cell, which are very few. Okay, here after DN2 stage, T cell commitment will occur. Oh. Okay, so this DN3 is committed cell, so that after this beta selection will occur. Beta selection means so that beta gene will completely rearrange on the surface of the double negative T cells and it will start next rearrangement if beta selection has done. So this question can also come ki at which step beta selection will occur. So after DN3 stage, beta selection will occur. So that alpha chain also will start rearrange after this step. So in DN4 step, TCR alpha locus rearrangement will also start occurring. Okay. Then our next step come, which will be double positive. Till this time, it was double negative. Here, both double positive, like CD4 plus and CD8 plus, both genes will present here. Double negative, then our double positive cell will come. Then its selection will occur. Either it should be CD4 positive or CD8 positive. CD8 or CD4. DN double negative, double positive, then single positive. So how this positive selection will occur? That either it should be CD positive or CD4 positive or CD8 positive. So it will occur by binding of these genes. Either it will bind to MHC1 gene, these TCR. Either it will bind to MHC1 or class MHC2. If it will bind to MHC class 1 gene, this receptor, then it will rearrange itself to CD8 positive cells. Or if the receptor will bind to self MHC class 2, then it will become to CD4 positive. So this we called as positive selection because this is self MHC restriction. It should bind to our self MHC. ठीक है. Other some cells will die by neglect. Some other will become other regulatory cells like Treg cells, Th17, IL. There are different kind of T cells. But if it is bind to MHC class 1, it will become to CD8 positive. If it will bind to CD4 positive, MHC class 2, then it will become to CD4. So how this development is occurring from double negative to double positive, then single positive. Next step come. So this positive selection is occurring in thymic cortex. One more important question. Positive selection is occurring in thymic cortex. Then next is negative selection. Negative selection will occur in thymic medulla, medulla region. This was occurring in cortex region. Next it will occur, negative selection will occur in thymic medulla. So here what will occur? If the interaction between T cell receptor and MHC class 2 is very strong, then those cells will negatively selected if the interaction between MHC class 1 or MHC class 2 and TCR is like not that strong, is weak, then that will be selected further. ठीक है? So if the interaction is very strong, then that cell will be negatively selected and will be die further by negative selection. Or if the interaction is low, then that cells will migrate from blood to peripheral tissues. So this is the story of the T cell development. I hope this is very clear to you. Like all the early thymocyte development from double negative to double positive to single positive, then positive selection and then negative selection. ठीक है, सो अभी हम अपने क्वेश्चन को देखते हैं. क्वेश्चन क्या था? टी सेल मैचुरेशन जो होता है थाइमस में वो किस तरीके से होता है. सो नाउ यू नो वेरी वेल, इट विल बी फ्रॉम डबल नेगेटिव टू डबल पॉजिटिव देन सिंगल पॉजिटिव. देखो, दिस इज द ऑप्शन दैट इज परफेक्ट. डबल नेगेटिव टू डबल पॉजिटिव टू सिंगल पॉजिटिव. ओके, मूविंग टू नेक्स्ट क्वेश्चन. Montauk's tuberculin test is related to which hypersensitivity? So इसके लिए भी हम पहले इस टॉपिक को समझेंगे. हाइपरसेंसिटिविटी का पूरा टॉपिक मैं समझाने वाली हूँ. ओके, ब्रीफली समझाऊंगी. ये जो टेबल है ये भी क्यूबी से है. इसको अच्छे से अगर कर लोगे तो बहुत अच्छे से आपका हाइपर सेंसिटिविटी वाला टॉपिक भी कवर हो जाएगा. लेकिन आपको ये वाला जो टेबल मैंने प्रोवाइड किया है ये पूरा याद होना चाहिए और मैं बताऊंगी इसमें से आपको क्या क्या याद करना है. Okay, so let's go about type 1. Type 1 is mainly related to allergy and atopy. In this, the main immune mediator is IgE antibody. So what IgE antibody will do? It will form, this is our allergen or antigen you can say, and this is our target cell. These are the antigen present. Sorry. This is our allergen. These are the receptor which will bind to antibody FC region of IgE. ठीक है, याद रखना यहाँ पे वेरिएबल रीजन से बाइंड नहीं कर रहा है हमारा एंटीबॉडी. हियर FC region of antibody is binding to the receptor and that will form cross linking. Allergen specific IgE, it is forming cross linking. ठीक है, not one antibody is binding to allergen, the two will form cross linking and that will bind to IgFCE receptor by their FC region. बस ये याद रखना. ठीक है, then this mechanism, this is the same that I was telling here. This will lead to degranulation. Okay, degranulation that will release the mainly enzymes like vasoactive enzymes that are like histamine. ठीक है, allergen and atopy. Typical manifestation, all these you can read. Then next is type 2. Type 2 is antibody mediated hypersensitivity. And what antibody involves here? IgG and IgM. Okay, IgG और IgM. So यहाँ पे antibody mediated hypersensitivity है. So antibody, these are our antibody. This will directed to the surface of antigen and it will lead to destruction of the target cell by either complement activation or other is ADCC, antibody dependent cell mediated cytotoxicity, जो मैंने अभी. Okay, so this type 2 hypersensitivity can manifested include manifestation of blood transfusion reaction, erythroblastosis fetalis, autoimmune hemolytic anemia. So these are very important points. ठीक है? So then next type 3 hypersensitivity, like this was antibody mediated only. This is immune complex, like antibody antigen immune complex will form. Like in this way, this is immune complex. Many antibody will form complex. In this case, only one antibody was there, but here immune complex will definitely be there and it can deposit somewhere in the tissues and that can further lead to complement activation. Okay, so Then this complement activation can like further activate neutrophil cells or whatever further phagocytic cells. Anyone can activate it, but this will be mediated by antigen, immune complex. Okay, and here important thing is Arthur's reaction, and also these rheumatoid arthritis, SLE, these things. These are the manifestations of type 3 hypersensitivity. Coming to type 4, like all these somewhere something related to antibody, but type 4 nothing has to do with antibody. This is totally independent of antibody, or say it is dependent on T cells. Okay, the immune mediator is T cell, and this is delayed type hypersensitivity. These are like just reaction will just like the our that something pathogen will enter and quick response will occur, but this will take some time to generate response because this is T cell mediated. Okay, like for antibody, memory cells are already there, but for T cells there will be interaction of MHC and T cells. After that the response will generate, so this will take some time. This is referred as delayed type hypersensitivity. Okay, so this can lead to either activation of macrophages or say phagocytic cell or either TC cells. TC is cytotoxic T cells. ठीक है? These manifestations are related to this type for contact dermatitis, tubercular lesions, and graft rejections. Okay, because this is related to T cells. T cells means self MHC restriction, and that is particularly related to graft rejection. I hope this slide is very clear to you. All these things are very important, as I already told. Key this diagrams, immune mediator, mechanism, and most important typical manifestations. This is also important, very important. Okay, now you can easily solve these questions. That Montauk's tuberculin test is related to IgE, not Montauk's tuberculin test like tubercular lessons. It is here. So tubercular lessons, or this is also related to tuberculin test. Antigen antibody complex. Yes, this is related to type 3, type 1, and antibody mediated is type 2. The only thing that is related to type 4 is antibody independent or say T cell dependent. Okay, so this question is for type 4 hypersensitivity. Okay, moving to next question, which cell is non-phagocyte? This is very famous question for all the entrance tests, whatever it is like DBT, GATE, NET, NII or any other exam. It usually comes in all the exams. So, and also this is very easy. Anyone can answer. As you all know, phagocytic cells include macrophages, neutrophils, and dendritic cells. These are all present in tissues, monocytes, eosinophils in bloods. So the options that include the phagocytic cells is macrophage, yes. Dendritic cells, yes. Eosinophils, yes. Basophils doesn't come under phagocytic. These are non-phagocytic cells. Okay, so the answer will be option D. I hope this is clear. So okay, so this slide till now I have I was having the idea of all the questions and related to their options and the topic, but now these slides I have just included so that you can have some idea of like 2-3 questions that I just randomly got in my Telegram group, but I don't have that much sufficient detail so that I can explain to you. Okay, so something question related to enzyme replacement therapy. Next one, what is the slope of horizontal line? So the answer will be 0. So mm, this is asking about horizontal line. This is our horizontal line, and they are asking about the slope. So see, if it will be like slightly tilted, then we can say there is something slope, but this is completely horizontal, so nothing slope will be there for horizontal line. Okay, if they ask about vertical line, so for vertical line, the slope will be undefined or say infinite. Okay, I got that one question was based on a plus b ka whole square, but about this also I don't have like much information. Ki what was it? Then the last question was, which is the weakest bond among four options. But I did not got four options, only one option I got is London dispersion force. Although itself it is weakest bond, but as I am not having the idea of other options, so I can't any comment that it will be the answer. Okay, so you can read more about the bond. So these were the questions that I got from the students on my Telegram group. Now you will be having some idea about this year itself in April 2024 NII exam, because some questions you got here and these are very easy question. If you have read like thoroughly about all the topics, then you can answer easily of all the questions. Or say you will get 60 questions, so you can answer at least 40 to 45 questions. If you can answer correctly this much question, then you can easily crack this exam. Okay, so what you can do these questions, and also as I said earlier starting in my video, that watch my, so as I was telling, you can watch my earlier videos also, starting from syllabus, as there is no defined syllabus for NII exam. So I have already made a video related to NII exam syllabus. You can watch that. And two or three NII PYQs related videos, and three are practice questions video, in which I have set up the questions which are related to NII exam, and also I have discussed the topic related to those questions also. So you can watch those videos also. And if you have any query related to these things or other things also, you can tell me on my Telegram group also, and personally on my Telegram, or either in comment box also. I will try to solve your all queries as soon as possible. Okay guys, so I hope you enjoyed in this video. Okay, so thank you so much for watching my videos. Please guys subscribe my channel. Let's do biology, and do like, share my videos.